Welcome, biology students, to the Dallas Independent School District's STEM Environmental Education Center. And we'd like to give a special welcome to the students at Woodrow Wilson High School. Thank you so much for joining us this morning for this virtual field trip. We wish you could be here in person, but since you can't be, we're going to do our best to make you feel like you're here during a virtual field trip experience this morning. Today, if you are watching this and have not registered for this field trip yet, you can still do that by going to www.tiny.cc/high school registration. Once you get there, you can get yourself or your class registered for this field trip. And we just use that information for our attendance purposes. And this morning's field trip is going to be all about genetics. During this virtual field trip, students will recognize that gene expression is a regulated process and predict possible outcomes of various genetic combinations, such as monohybrid crosses, dihybrid crosses, and non-Mendelian inheritance. So we're going to start things off this morning by exploring gene expression with Mr. Monroe. Next, we will look at monohybrid crosses with Mr. Mirez. Third, we will look at dihybrid crosses with Ms. Schramm. And last but not least, we're going to explore non-Mendelian inheritance with Mr. Dominguez. While we're doing all of that, if you have any questions, you can ask those, but since this is a virtual field trip, the way you'll ask a question is you'll need to go to www.tiny.cc/question-answer to fill out a super short form and submit any questions you have for us related to genetics. You can ask as many questions as you like, and we'll do our best to answer all of them in the time that we have with you this morning. Now let me stop sharing my screen here and turn things over to Mr. Monroe, who's going to get us started with gene expression.
Okay, good morning, students. My name is Mr. Monroe, and we're going to be exploring part of a very deep subject called gene expression. And this program, really, I wish I could do it in completeness, but it would take much more than the time that I have or I've been allowed to be with you. So basically, I'm going to start out by talking about what gene expression is. It's the process by which information from a gene is used in the synthesis of a functional gene product and enables it to produce end products, protein or non-coding RNA, and can ultimately affect phenotype as a final effect. The process of gene expression is used by all known life, eukaryotes, including multicellular organisms, and prokaryotes, such as bacteria and archaea, and utilized by virus to generate the macromolecular machinery for life. Now, this process is strictly regulated. Regulation of gene expression is the control of the amount and timing of the appearance of the functional product of a gene. Control of expression is vital to allow the cell to produce the gene products it needs when it needs them. In turn, this gives cells the flexibility to adapt to our variable environment, including external signals, damage to the cells, and other stimuli. More generally, gene regulation gives the cell control over all structure and function, and is the basis for cellular differentiation, and versatility and adaptability of an organism. Now, gene expression happens internally, the internal environment, and it happens externally, the outside environment. Humans, for example, have an organ that is called the liver. And its job is to remove the toxic substances like alcohol from the bloodstream. To do this, the liver cells, gene expression, encoding, submits pieces of enzyme called dihydrogenase. This enzyme breaks alcohol down into a non-toxic molecule. So that's one example of gene expression happening within an organ that is found in the human body. Also, if we look at an example from the outside that's actually influenced by temperature. which is part of the outside environment, we can look at the Himalayan rabbits. And the Himalayan rabbits basically are white in color. And let's take, for example, gene C. Gene C controls pigmentation for the Himalayan rabbits. Because this gene is active when the environment temperatures are between 15 and 25 degrees Celsius, the rabbits that are reared in 20 degrees Celsius temperature has pigmentation on the ears, the nose, and the feet, where its body loses most of the heat. The rabbit that's reared in temperatures above 30 degrees Celsius has no fur pigmentation because gene C is inactive at these higher temperatures. So that's an example of gene expression dealing with the external environment, the outside. And as you can see, my little drawings here, we can see that these guys, this guy here would be the one that is reared at 20 degrees Celsius or less. So the tail is dark, the feet are dark or black, and that's where the body loses most of the heat. So that's an adaptation that is caused by gene expression. I also have a live animal that I want to share with you guys today. Her name is Bessie, and Bessie is a Curtonic quail. Let's see if I can catch her and get her out. Come here, Betsy. Now, Betsy is a very popular quail that is raised by a lot of people simply because of gene expression. And the gene expression that's going on with this species of quail is that it has an acceleration of maturity. It takes only about 7 weeks for this species of quail to become an adult. And so because of that short period of growth, then they can reproduce at a very early age. And a lot of people raise these for a business simply because of that. So that's one example of gene expression. Now I'm going to put Bessie back. And let's talk about another example of gene expression. You know, there are organisms that sometimes are, in fact, I will say the group that we refer to as reptiles, they are known to be where they can, how would you say it? I guess the best way to put it is TSD. And what that is, temperature determining the sex of that organism. TSD stands for temperature sex determination. And alligators have been found to be one such animal that will go through that. And according to research, at a certain stage in the embryonic development, if the temperature is 33.5 degrees Celsius, it has an effect on the embryo to determine that it's going to be a male. Now, if it's below 30 degrees Celsius, that's going to help determine whether it, at that particular time, when I guess what they call it is TSP, temperature sensitive period, it will determine that most likely it's going to be a female. Now, that has some ramifications because if we look at the world today, we're going through what they call climate change. Things are getting warmer. And if you really think about it, someday we may have more males than we have females. So what will that have? What will that do to impact that particular species or that particular animal, the alligator? That's something to think about, guys. Now, hopefully I've given you kind of an overview about gene expression. I could go on and on. I could put diagrams up, but I'm about out of time. So what I'm going to do is turn it back over to Mr. Mr. Broughton, so that if any of you have any questions, I bet he can answer those for you. You guys have a good day today and a good rest of the virtual field trip.
Thank you, Mr. Monroe. The question that came in was, what are the steps of gene expression? And there are two major steps, transcription and translation. During the process of transcription, the information stored in a gene's DNA is passed to a similar molecule called RNA in the cell nucleus. And then during translation, the second step is getting from a gene to a protein, and that takes place in the cytoplasm. All right, now we're going to explore monohybrid crosses with Ms. Ramirez.
Hello, my name is Ms. Ramirez, and in this segment, we're going to be learning about monohybrid crosses. Before we start, though, I do have an animal friend that's going to help introduce that topic. So I'm going to give you some clues to who this animal friend is, and then I'll let you guys guess. So this animal friend is a lagomorph, meaning that he has two pairs of upper incisors. Those are simply teeth. This animal is a prey animal. This animal is also an herbivore. This animal belongs to class Mammalia, and this animal can run and jump rather easily. So go ahead and make your guess to who this animal friend is, and I'll go pull him out. This is Baby Bunny Lucky. He's rather energetic this morning. So Baby Bunny Lucky is probably about 5 months old now, and I actually know his parents, so he's a great example of what genetics can do. So I know what his mother looks like, and I know what his father looks like, and he actually looks a whole bunch like his father. He's also very fidgety right now. So I'm gonna go ahead and let him play on the floor, but we're gonna take a quick little examination about rabbit genetics as well. So let me go let him play with his brother, and I'm gonna share my screen with you guys, and we'll get that started. So let me get that screen share going. So for our presentation today, I have a couple of questions for you guys that I would like for you to keep in mind. And as I'm reading the questions on the right, you'll see a little video with my rabbits. So your essential questions for the day are: 1, what is a monohybrid cross? And 2, what are the genotypic and phenotypic ratios, and how are they calculated? So hopefully you should be able to answer those 2 questions by the end of this session. So on the right, you'll see the video. So that is the mom, that's Mochi. Here's a picture of the dad and the mom together. And so what do you think the offspring will look like? And then here's a little video of the litter that they had. So Mochi had 7 babies. Baby Bunny Lucky was one of those. And that is what he looked like when he was a few weeks old. He was very pudgy. This is the bunny family. So you can kind of see the resemblance in the coloration as well. And I think that Baby Bunny Lucky most closely resembles his dad, Clover. They have that same fur color. Mochi has a very unique coloring. The mom, half her face is black and the other half is brown, and she has a kind of like a mix of black and brown patching on her fur color. And it is interesting to really do some deep research into rabbit genetics. Breeders use it a lot to help them create different colorations. So our next slide that we're going to take a look at are just some quick vocabulary words that are important to help us understand our lesson today. So teachers, feel free to pause the video so that students can test their prior knowledge, see if they can fill in the blanks with the correct vocabulary words. But I'm going to go ahead and jump right into it. So question number 1 is: a blank is a form of a gene. A Punnett square illustrates how the parents' alleles may be passed to an offspring. So for number 1, an allele is a form of a gene. And we're going to be looking at examples of alleles as we complete our Punnett squares today. So again, an allele is just a form of a gene. Question number 2, a blank of an individual describes the specific alleles of a gene that the individual carries. This is also known as the genetic makeup. And hopefully for number 2, you guys said the genotype. And that's easy to remember because genotype starts with a G and genetic starts with a G. So the genotype is just the genetic makeup of an individual. Question number 3, the blank of an individual describes the outward expression of the genes carried by an individual. So if the genotype is the genetic makeup, what do you think the answer is for number 3? And hopefully you guys said the phenotype. And an easy way to remember that is the phenotype is simply the physical appearance of an individual. And phenotype and physical both start with the same letter P. So again, phenotype is just the physical appearance. And then number 4, blank is considered the father of genetics. I have an image of him right over here on the right. Hopefully you guys know him because he did a variety of experiments studying pea plant genetics, and that is Gregor Mendel. So those are a couple of vocabulary words that will be helpful throughout this presentation. So in our next slide, we have a couple more vocabulary words. So our first one is homozygous. Homozygous individuals are simply those individuals with 2 copies of the same allele. So remember, an allele is just a form of a gene. So when we look at our Punnett squares, if you see 2 uppercase letters or 2 lowercase letters, those would be homozygous individuals. Then we have, on the other hand, we have a heterozygous individual. Those are individuals with 1 copy of one allele and then another copy of a second allele. So they have 2 different copies. An example of a heterozygous individual would be if you see the genotype capital letter and then a lowercase letter. So that would be a heterozygous individual. So homozygous means the same, heterozygous means different. So using that explanation, look at our image over here on the right. Are the parent pea plants homozygous or heterozygous in this diagram? And how do you know that? And looking at our parent pea plants, we can see that the yellow parent has 2 lowercase g's, so they're the same. And our green parent has 2 uppercase g's. So again, they're the same. So in this example, both parents happen to be homozygous individuals. They have 2 copies of the same allele. Some other words, we have our dominant allele, and a dominant allele is the one that is expressed in a heterozygous individual. An easy way to remember that is dominant alleles will be capitalized. On the other hand, we have a recessive allele. Recessive allele phenotypes are only seen in homozygous recessive individuals. So a recessive allele is often denoted in a lower case. So for example, if we take a look at our picture here on the right, what allele do you think is the dominant phenotype? And hopefully you said that the dominant phenotype is going to be green color because it has the capitalized letter. So again, the dominant allele is going to be capitalized and the recessive allele is going to be lowercase. And again, here on our example, we have the yellow is lowercase, so we know that that's going to be the recessive, and the green is uppercase, so we know that that's going to be the dominant trait. Now let's look how we can apply those alleles to a Punnett square. So we're going to be examining monohybrid crosses, and that is a genetic cross prediction using a Punnett square, but we're only going to be using 1 trait. So in the next segment, you're going to be looking at dihybrid crosses, and Ms. Schramm will discussing 2 traits. In this one, we're just doing 1 trait at a time. So here is our Punnett square, and we're going to be doing a monohybrid cross. This is an example using our longhorn Freckles, who's the dad, and our mom, who is an Angus. Her name is Obsidian. So we're going to see what is the probability of having offspring with spots or no spots. So take a look at our little key over here. Capital S means spots, lowercase s means no spots. So which one is the dominant trait, and how would you know that? So given the information from the last slide, we know that spots is the dominant trait because it is capitalized. No spots is the recessive trait because it's lowercase. In this case, for our example, we're going to say that the dad, Freckles, is a heterozygous individual because he has a capital S and a lowercase s for his genotype. So because he has 2 different alleles, he is heterozygous. Now let's look at the mom. The mom has 2 lowercase s's. So she is homozygous because she has 2 of the same. Now we're going to see what happens when we cross the two. So to complete our Punnett square, it's super simple. We're just going to do a cross. So we're going to work on our first quadrant right here. We're going to combine this uppercase S from the dad and the lowercase s from the mom. And that is going to give us capital S, lowercase s. We're going to move to the next quadrant. We're going to take, again, the capital S from the dad, combine it with the lowercase s from the mom, and that will give us uppercase S, lowercase s. Move on to the next quadrant. We're going to take the small s from the dad, combine it to the small s with the mom, and now we have two lowercase s's. The last quadrant, we're going to combine again, take the lowercase s from the dad, combine it with the lowercase s from the mom, and that will give us two lowercase s's. So now we're going to determine the phenotypes and the genotypes for the offspring. So again, phenotypes means what will the offspring look like? Will they have spots or will they not have spots? Using our answers from our Punnett square, we can see that 50% of the offspring will have spots. And we know that because in order to have spots, we have to have two lowercase s's. So look at our key, the lowercase s means no spots and the uppercase s means spots. So 50% of offspring will have spots. So this is our dominant right here, two uppercase s's and another uppercase s. So half the offspring will have spots. The other half, look on our other side of the Punnett square, these two squares have lowercase s's, so they will have no spots. So 50% will have spots and 50% will not have spots. And if we put that in a ratio, we can say it's 2 to 2. So 1, 2, there's 2 for spots, and 1, 2, 2 for no spots. So that's a ratio of 2 to 2. Now let's look at the genotypes. Genotypes means the genetic makeup. So 0% will have homozygous dominant. So we don't have any that are uppercase S, uppercase S. So that's why we have 0 right here. For the recessive traits, homozygous recessive, the lowercase s's, look on our little chart over here. We have 2 that will be homozygous recessive, meaning 2 lowercase s's. So we can say that that is 50%. The other is heterozygous dominant. So we're going to be looking for the number that has an uppercase s and a lowercase s. So we have 2 of those right over here. So that's 50%. So if we wanted to do a ratio with the genotypes, we could say it's 0 to 2 to 2, because we have 0 that are dominant homozygous. We have 2 that are recessive homozygous, and we have 2 that are heterozygous. So that's how we get those ratios. And then here's just a quick review. Here's our Freckles. He mated with Obsidian. They had a baby, and this is baby Oreo. And baby Oreo is a longhorn, and he happened to have spots. And my challenge question for you guys is to complete the Punnett square. These are 2 chickens and roosters that we have here at the EEC. This is my blue silky pepper, and this is Prince, the naked neck rooster. If they were to mate, see if you can complete the Punnett square to figure out the ratios, the genotypic ratios and the phenotypic ratios. And again, feel free to pause the video to allow time to do that, but I'm going to move right along. And the last slide, I just wanted to show you guys these interesting chickens. These are the naked necks. This naked neck happens to be homozygous for the naked neck trait, and we can see that because it's 2 capitalized letters. This naked neck, look how it's a little bit different. This one is heterozygous for the naked neck trait, so it has a capital N and a lowercase n. And notice that when it's a heterozygous individual, it still has a little puff of feathers around its neck. So I'm going to stop my screen share, and that is all I have for you guys for genetics. We're going to give it back to Mr. Broughton to answer any questions.
Thank you, Mr. Ramirez. The question that came in was, when was Oreo born? And she was born in December of 2019, I believe, if I remember correctly. All right, now we're going to explore dihybrid crosses with Miss Schramm.
Hey everybody, it's Miss Schramm, and I also have a bunny friend to introduce you to, and his name is, Peanut Bunny. Oh, he's so sassy. Peanut, it's okay, Peanut Bunny. You've been on camera before. Okay, so Peanut Bunny, he's going to be the hypothetical bunny daddy today. So thank you for that, Peanut Bunny. Sorry to disturb you. He's kind of grouchy this morning. Sheesh. All right, so let's get going. Presenting. All right, so at the end of my little section, you'll be able to predict outcomes of dihybrid crosses. So like you learned with Miss Ramirez, monohybrid crosses are used to predict the outcome of just one trait. Dihybrid crosses are to predict two traits. So right here, it's the same diagram as she showed you before, the Punnett square about green pods or yellow pods. So that is predicting one trait, the color. Then on the right, it's a Punnett square with 16 boxes, and they're predicting whether the peas are going to be round and yellow, round and green, wrinkled and yellow, or wrinkled and green. So that is predicting two different traits, the color, or sorry, the shape, and if it's wrinkled or not. Okay, so like I said, we're going to use Peanut Bunny as the hypothetical bunny daddy, and we are going to try and predict him mating with another bunny and looking at two traits for tall ears or floppy ears and brown fur or tan fur. So this is hypothetical, so I put a little asterisk by it. In rabbits, brown is dominant to tan fur. So for our purposes, we're going to assume that brown is dominant to tan fur. And then also in rabbits, which is true, is long straight ears are the dominant to floppy ears. So that picture to the right is showing a bunny with, well, actually that's Ms. Ramirez bunny now, Pfeiffer, when he was a baby, and his ears are pointing up. So that is a dominant trait. So the letters that represent the genotypes and the phenotypes of the rabbits. So capital BB is going to be brown fur. Then uppercase B, lowercase b is going to be brown fur as well, because remember the capital B, that's brown fur, is dominant. Then two lowercase b's is going to be tan fur. Then double capital E's is going to be tall ears, which is dominant. So capital E and a little e is going to be also tall ears. And then two lowercase e's are going to be the floppy ears, and those are called lopped. So meet our bunny parents. So Peanut Bunny, who you've met, has brown fur and tall ears. So for our purposes, I just made up his genotype being capital B, lowercase b, capital E, lowercase e, to make it a little more interesting. So he is heterozygous, theoretically. And Bun on the right, who belongs to Mr. Dominguez, is a Holland lap bunny. So she has the flap down ears and she is a tan rabbit. So her genotype is going to be lowercase b, because tan fur is recessive. And then lowercase e, because floppy ears is recessive. So Peanut Bunny, his genotype, I just kind of made it up for our purposes, but Bun's is pretty much accurate. Okay, so dear goodness, hang in there, children, because now we have 16 squares on our Punnett square. Whew, but we can do it. So we've got Peanut Bunny and his genotype on the top, and Bun, who is the potential mother, on the side. So I have all these question marks. What on earth are we going to put there? Because now there's two traits and we're all scrambled like eggs. So let's check. We are going to use the FOIL method. Now this is a lot more simple than it looks, so don't worry. So with the FOIL method, you are going to go with the first trait, then the outside, the inner, and the last. So focus right now on just Peanut Bunny. And I have Peanut Bunny's genotype right here for the cross. So we have the fur and then we have the ears, right? So for Peanut Bunny, we are going to go with the first letters. So we've got capital B, capital E. So that's what's going to go first. Then we have capital, or outside, capital B, lowercase e. That's going to go next. Then we have lowercase b, capital E. That goes next. Then lowercase b and lowercase e. That goes next because that's last. So we went with the first letters, the outside letters, the inner letters, and then the last letters. Whew, and we made it. So Peanut Bunny's was complicated because he was heterozygous. So you really have to make sure that you're following the FOIL method with that one. Now Bun is a little easier because she only has the recessive traits. So all of her letters are going to be lowercase. So while you can use the FOIL method, you already know the outcome is going to be all lowercase. So you really don't need to go through the FOIL method, but in case you feel more secure doing it, that's fine. And your results are going to be the same. So all of Bun's are going to be B, E, all lowercase. Simple. So that's not too bad. So let's pop those into our Punnett squares. All right. So once again, Peanut Bunny's is on the top and Bun's is along the sides. And now we start to fill. And it's not so bad. Let's try it. Okay. So when we fill these up, we are just going to go from the top column to the second row. So we've got capital B, capital E, and then add that to a lowercase b and a lowercase e. So all you have is one of each, that's easy. And since Bun is homozygous, it's very easy because we just add everything the same, all columns. So this combination is going to be the same here, 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 and here. So that whole column is going to be the same because all of hers are the same. So now we go to the next one. Capital B, lowercase b, lowercase e. So it's all going to be one capital B, one lowercase b, and two lowercase e's the whole way down because these are the same. So once again, we go to the next column. We have a lowercase b and another lowercase b, an uppercase e and a lowercase e. And it's going to be the same all the way down. Then we've got a lowercase b, a lowercase b, a lowercase e, and a lowercase e. So that's going to be all lowercase all the way down. So even though it looks like a lot of work with 16 boxes, it's really quite simple. You just have to make sure you're following the squares and kind of taking your time and double checking your work. So let's go predict the outcomes. Let's check our results. So once again, this looks like a mess and it looks overwhelming, but what's on the left is just the Punnett square we've already filled out, and that's for reference. And then lower below here, I put a little reminder so we can remember what each of those genotypes stand for the phenotypes. So what's this gonna look like? So because this is dihybrid cross, remember we're trying to predict two traits. So let's focus on the first question. How many offspring have brown fur and tall ears? All right, so we know brown fur is either going to be uppercase B 2 times or just one uppercase B. Whew. Okay, so let's look. And then we also have tall ears. So we know it can be uppercase E or uppercase E. So let's go ahead and look. These ones, this first column, remember they're all the same, so that helps. This column has uppercase B. So I know this is brown fur. And I see an uppercase E. So that's also tall ears. So I know I can count these columns because remember, these are all the same. So I know I have 1, 2, 3, 4. All right, let's see if I have any other ones. So there's 4 so far. In this column, I have brown fur because I see capital B and a lowercase b, but it does not have tall ears because it has 2 lowercase e's. So I cannot count that column. So this one has tan fur, so I can't count that column because I'm looking only for brown fur. Then the next one has tan fur again, so I'm not even gonna look because I'm looking for brown fur and tall ears. So the answer to the first question is 4. These ones are the only bunnies that have brown fur and tall ears. All right, so the next question, how many offspring have brown fur and floppy ears? So remember, that's capital B, capital B, capital B, lowercase b, and then we need floppy ears, which is the double lowercase e. All right, this one has brown fur and floppy ears. Okay, so I can count that column. 1, 2, 3, 4. Boom, easy. So that is 4, and let's check this next row. Or sorry, column. So I see tan fur. Well, I can't count that because I'm looking for brown fur and floppy ears. So this one has tan fur, we're not counting that. And once again, this has tan fur, so we're not counting that. So we just have 1, 2, 3, 4 with brown fur and floppy ears. I'm noticing a pattern here. So next, I have the question, how many offspring has tan fur and tall ears? So we know we need to find lowercase b, and we're gonna need at least one uppercase B for those tall ears, the dominant trait. So I'm looking at this column and I have tan fur, tall ears. So that means 1, 2, 3, 4. If I look at the next one, it has tan fur, but floppy ears. So that's not what I was looking for. So I'm gonna have 1, 2, 3, 4 with tan fur and tall ears. Then, as you may have guessed, noticing the pattern, how many offspring have tan fur and floppy ears? Well, sure enough, the recessive traits for tan fur and floppy ears is right here. So that is 1, 2, 3, 4. All right, so there's our answers. There's 4 of each. Since there are 4 of each, out of the 16, 4 offspring would have brown fur and tall ears like this first bunny. That is 25%. Then 4 offspring would have brown fur and floppy ears. That's another 25%. Then 4 offspring would have tan fur and tall ears. That's 25%. And 4 offspring would have tan fur and floppy ears. That's 25%. So the ratio is 1 to 1 to 1 to 1. Boom, simple. So 25% chance that you're gonna have one that looks exactly like Peanut Bunny, and 25% chance that you will have one that looks like the Mama Bun. Simple as that. So my last question before I send you off is, I have a bunny mystery. So Salem is a bunny that has one ear that stands up straight and tall and one ear that is locked, so it flaps down. How can this be? And I'm gonna let you get a good look at him before I send you away. Come here now, Salem bunny. Can you be lovelier? Yes, so you don't be so drama. So you can see, well, right now he's a little like sassy because I just pick him up. But usually this ear stands up straight and this ear flaps down. So how could this be? All right, thank you so much for joining us. I hope you have fun and I'll see you next time.
Thank you, Ms. Schramm. The question that came in was, is there such a thing as a tri-hybrid cross? And yes, there is. So there's a picture of one, and that is for examining 3 traits. So in this example, it's pea plants again. The R stands for round or wrinkled peas. The Y is for yellow or green. And then the S is for a smooth pod or a wrinkled pod for looking at the whole pea pod and not just an individual pea. And there's even tetrahybrid crosses, which are for 4 traits. So there's, it can go on and on. But now we're going to look at non-Mendelian inheritance with Mr. Dominguez.
Hey guys, in this portion of your virtual field trip, we are going to be talking about non-Mendelian inheritance, and we are going to focus on codominance and incomplete dominance. And I wanted to show you guys what I think is a pretty awesome example of codominance. So these guys are isopods. You guys see them crawling around. So when I first got these, I had a batch of white and black isopods. These are the Scaber variety of isopods, and they're all the same species. And when I originally bought them, so I only had black and white ones. Now, after a couple of years of them being in this tub and breeding, I ended up getting a bunch of these color variations. So I had a bunch of well, this is about as close to white as I used to have, and this is about as close to the original black that I had. But after a while, they started mixing their colors. The genotypes for the white color and the black color started expressing themselves in a very codominant fashion, meaning that both the black and the white color, those genes were being expressed in their phenotype equally, or some of each of the color was being expressed. And now what we have is a very awesome looking isopod that looks kind of like a cow. So we got some black and white. Now there are some where we have more of the black being expressed, but you can still see some white, and you see some black here as well on this mostly white one. But I thought this was a really cool example of codominance. So both colors are being expressed. So I don't think I have many of the all white or all black ones left. So even this one that is mostly white has some black speckling. So I thought that was a really cool example of codominance. And remember, codominance is when genes, in this case for color, are expressed as a mix. So you're going to get both black and white colors being expressed in the phenotype. All right, guys, to get a better understanding of non-Mendelian inheritance, we have to understand the three types of dominance. So there is complete codominance and incomplete dominance. And we're going to use an example with flowers. So we're going to have three different scenarios, and we're going to be talking about flower color. So we're going to have a red flower, and we are going to have a blue flower. And we're going to have a red R for the red allele or the red phenotype, and a blue R over here. And we are going to have three different scenarios where we're going to have three different genotypes expressed, and we're going to see what phenotype you would see. Now, let's say, and we're going to talk about what would happen in each of these types of dominance, so complete codominance and incomplete dominance. So let's say that you had a genotype that looked like this. You had red R. So that genotype you would expect in complete dominance or regular old Mendelian inheritance. You would have a red flower, correct? Because the only alleles expressed in this genotype would be for the color red, and you would also expect the same thing from codominance and incomplete dominance. You would have that red color expressed. Now let's say you had blue R. Well, you would have the same scenario in each of these types of dominance. You would have a blue flower, and a blue flower. Now, where it gets interesting is when you have the heterozygous genotype. So let's say you have a red R and a blue R. Now, let's say that the red allele was dominant, and your work with a Punnett square would tell you that, well, if the R is dominant, well, then you would have a red flower, right? So you would have a nice, beautiful red flower, even though both alleles are expressed. If red or red R is the dominant allele, you would expect red as the phenotype, correct? Now, in codominance, it gets a little bit more interesting because in this scenario, both alleles would be dominant and both would be expressed. So you would have not a red flower, but you would have an interesting flower. You would have a red you would have a red-blue flower. So you would have something that maybe looks like this. So both alleles in this genotype would be expressed in the phenotype. So you would see both colors. So something that maybe looks like this. So there we go. So you would get a flower that has both alleles expressed in the phenotype. So now in incomplete dominance, neither allele or in the genotype, in the heterozygous genotype, is expressed in the traditional way. So you would get something that is maybe in between. So you would maybe get a purple flower here in this instance. So you would get something that maybe looks like this. So something that is in between the red and the blue allele. So this genotype, in this instance, you would get a purple flower expressed in the phenotype that you'd see. So, and we see this happen all the time. So if you go into a flower shop or maybe your own garden, sometimes you will get cross-pollination, and you will get instances of a red flower and a white flower. So let's say a red rose and a white rose getting you a pink rose. So that is incomplete dominance, and both codominance and incomplete dominance are examples of non-Mendelian inheritance. All right, guys, now that we have a better understanding of what incomplete dominance and codominance is, and just remember that those are two examples of non-Mendelian inheritance, I want you guys to take a look at this little example of possible incomplete dominance. So a long time ago, my dog Princess and my dog Higgs had a baby, and this is her, that's Olive. And she is showing what is considered a blue coat color. So it's neither her blondish kind of golden color or his black fur color. So she is something in between. So I wanted to ask you guys, do you guys think that this is incomplete dominance or not? Well, it's not, and we have to be careful because genetic expression is very nuanced. In fact, the blue coat color is a mutation in the alleles that express fur color and skin color in dogs. So we have to be careful when we are talking about genetic expression because it is a very complex topic. But I hope that today you at least came out with two examples of non-Mendelian inheritance from this presentation. And I hope that you guys have a wonderful rest of your day. And I will see you guys next time for the next virtual field trip.
Thank you, Mr. Dominguez. I'm going to share my screen here to do a quick recap of what we covered this morning. So again, today's virtual field trip was all about genetics. During this virtual field trip, students recognized that gene expression is a regulated process and predicted possible outcomes of various genetic combinations such as monohybrid crosses, dihybrid crosses, and non-Mendelian inheritance. So we started off by exploring gene expression with Mr. Monroe. Next, we explored monohybrid crosses with Mr. Ramirez. Third, we explored dihybrid crosses with Ms. Schramm. And we just finished exploring non-Mendelian inheritance with Mr. Dominguez. Thank you so much for joining us this morning. We appreciate you taking the time to learn about genetics with us. We hope you enjoyed this virtual field trip, and you can let us know whether you did or not by going to www.tiny.cc/hsfeedback and filling out a short feedback form for us. I hope to see you again for our next virtual field trip for biology students. Have a great rest of your day, and we'll see you next time.